I'd like to begin by acknowledging the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation, and pay my respects to their elders past, present, and the elders of any other communities that may be here today. So welcome everyone. We've got Professor Geoff McFadden come all the way from across the road to talk to us today. He is the first of a reinvigorated divisional seminar series, which I tasked the a group of postdocs to start to bring in some external speakers for us to listen to. Geoff is an evolutionary biologist turned accidental parasitologist. And the word accidental parasitologist is his words, not mine. He worked on, when I met him, he worked on the evolution of chloroplasts until one day he found himself doing a sabbatical in Nova Scotia, looking for something to work on. And I'll leave the rest of the story to him to tell you one day. It was Geoff that discovered the apicoplast, and that is the apicoplast that is named after the phylum and not the phylum that's named after the apicoplast. So Geoff has had a stellar career, the discovery of the apicoplast, lots of stuff before and after. He is a fellow of the Australian... He's the fellow of the Australian Academy of Sciences, and I would consider Geoff to be the golden child of the ARC. He's been a Federation Fellow and a Laureate Fellow as well. So without further ado, I think Geoff's going to tell us an interesting story about malaria, the origin and elimination, and I just welcome Geoff to the stage.
Thanks, Chris. It's kind of nice to be introduced by a former student now as director of the division. We'll get right into it. We're going to start here about symbiosis, which is living together. And this can happen in a couple of different ways. At one extreme is cooperation of both partners, which is a win-win situation. And the classic example is the mutualism that is coral reefs, and I'll talk a bit about that. At the other end of the spectrum is where one partner exploits the other partner, and you have a win-lose situation, such as parasitism and obviously malaria. So the origin of malaria probably looked something like, did someone ring a taxi or something?
Just ask people to mute themselves.
Yeah, could you mute yourselves? This is one of Patrick's photos. So this symbiosis, this mutualistic cooperation, involves an animal, a cnidarian on the right, and a symbiont, the dinoflagellate, on the left. These are algae that photosynthesize, and the way the partnership works is the algae photosynthesize, they give glucose from that photosynthesis to the animal, and then the animal provides them with a source of nitrogen, shelter, and carbon dioxide. And so they live inside the animal cell, inside a vacuole inside the animal cells. They provide as much as 95% of the animal's nutrition. So these animals can eat, but they largely live from the glucose of their symbionts. And it's a closed system. So the nutrients go round and round inside the animal and the system. So it's very high productivity, even though the waters are fairly nutrient poor and often beautifully clear. And when this symbiosis breaks down, that's what you see when you have coral bleaching. I'm not going to go into much of that today, but what happens is the animals throw out their symbionts. They eject them because they're running too hot for the photosynthesis, and then the coral is bleached. Dinoflagellates are, I think, the coolest organisms on the planet. They have 3 times more DNA than you do. They have about 50 copies of every gene, and all copies are expressed and all copies are different. So if you think about that from a Darwinian perspective, it's just ridiculous. They have unique base modifications, so they have unusual DNA, permanently condensed DNA, and they actually, Sebastian Gornick in Ross's lab and my lab figured out that they don't have histones. They're the only organisms that pack their DNA using a different protein. This is some video of several different dinoflagellates paddling around that my PhD supervisor made, and the one in the middle, that crescent-shaped one, is totally gorgeous, but it can turn into those little motile ones. So when this symbiosis is good, it's pure bliss. So this is a dinoflagellate symbiont in this jellyfish in Palau, up near the Philippines. And in these lagoons, in these islands, literally millions of these jellyfish. You can see in the centre panel there, and you can swim amongst them. They don't have any stingers. They don't even eat. So these jellyfish are totally photosynthetic. And during the night, they swim down to the western end of the lagoon, waiting for the sun to come up. And then during the day, they progress down to the eastern end of the lagoon to catch the last rays of the sun. And then in the next night, they swim back again. So there's this beautiful symbiosis between animal and plant to create that. And then, of course, that can break down in these corals, which have ejected their symbionts because the water's got too warm and the photosynthesis was running too fast and they were creating too many oxygen radicals, probably. They chucked them out. These corals aren't dead. They are still alive, but they've just lost their colour, which is from the algae. And they could survive if they could get the algae back. And Madeline Van Oppen in our department is working hard on figuring out how that might happen. So I have started off with a question of how do the dinoflagellates actually give glucose to their host? And these dinoflagellates are interesting because they can live inside the animal, but they can also live independently, free living in the seawater. And so I figured out there must be some kind of transporter to export the glucose from the symbiont to the host. We decided, rather than doing this on corals, we decided to do it on anemones because they're much easier to grow, they're a better system. But jellyfish are anemones are basically upside down jellyfish, and corals are basically a whole bunch of anemones embedded in a rock that they built for themselves. So these anemones turned out to be a really good model. They can reproduce sexually and asexually. You can clone them. You can grow them without the symbionts, so you can bleach the symbionts, but you have to feed them a lot of sea monkeys. You can reinfect them with different symbionts, and we've done a lot of work on why they take particular strains and not others. We can talk about that today. And we can grow the symbionts separately, obviously. So we started off with two questions. How do they transfer the glucose to the host? And once we figured this out, it actually then opened up a really interesting concept, which I'll tell you today about, did this transfer actually pave the way for parasitism? So this started off with Karen doing a transcriptomics project with simple concept. We would take the dinoflagellates when they're free living in culture and photosynthesizing versus dinoflagellates that have been living inside these anemone hosts on the right there, and just basically compare the expression of different genes. And Karen identified a range of different genes that were upregulated in the symbiotic relationship when the dinoflagellates were inside their hosts, and a number of different transporters. And one that sort of jumped out at us is a transporter called SWEET. And SWEETs are a kind of transporter that were discovered in plants about 13 years ago. They are a 7-transmembrane domain transporter, and they're facilitators. So they're passive transporters, and they simply facilitate diffusion down a concentration gradient, and they're uniporters, just a single substrate. It turns out SWEETs are everywhere. All organisms have them, and the substrates that are known thus far are glucose, fructose, and sucrose. So it was an intriguing option that this was upregulated when the organism was in a symbiotic relationship. So in plants, SWEETs export glucose from the photosynthetic cells. That then goes into the plumbing system that plants have, and it's transported down to feed the roots, which obviously can't photosynthesize, so they needed a source of nourishment. And I sort of like this idea because you've got a photosynthetic tissue, exports glucose to a tissue that's heterotrophic, such as the animals surrounding, in this case, the roots of the plants. They also use SWEETs to put sugar into nectaries to attract bees, which is nice, and make honey. They also use these SWEETs to load sugars into seeds for starch to give the developing seed embryo a go. So SWEETs are transport facilitators, and they basically can send glucose, or allow glucose to pass through the membrane down its concentration gradient. So glucose goes from a source of high concentration to a source of low concentration. They're simple facilitators. So the question was, do they use these SWEETs as a transporter to feed the glucose to their host? And we tackled this in a couple of ways. Is it a canonical SWEET? Because no one had ever looked at a SWEET in dinoflagellates before. Where is it located? The hypothesis was that it would be in the cell membrane of the dinoflagellate, allowing the glucose to leak out from the high concentration gradient. And it would be expressed probably when they're in symbiosis, like we've seen in the RNA studies, because it wouldn't make any sense to be exporting glucose out into the ocean when you're not living inside a host, and figure out what the substrate was of glucose. So this is my first adventure in AlphaFold. This is of striking for an evolutionary biologist to see two proteins that are about a billion years apart, still have the purest kind of structure. We notice that these extra helix on the dinoflagellate suite, we don't really know anything about what that may or may not do. So we built an antibody to that protein, and the antibody worked rather nicely, picked up a nice clean band here, and Kieran's labelled a bunch of these dinoflagellate cells here. So here's their nuclei. This is the sweet label. It's around the periphery of the cells. This is their chlorophyll. These are just the mergers. And so it's just that the sweet protein is around the edge of the dinoflagellate. As I stand here, the cell, just cellulose, it's like the cell, and then you can see that the sweet protein is just adjacent to the cell. There's the red fluorescence from the chlorophyll. If we just look closely, you can see the cell edge. And that's the sweet staining. So if you back and forth, you can see that. If I'm giving a tick attack. So Kieran then wanted to check, is this sweet protein really more abundant in the host situation, in the symbiotic situation? And so did this with the antibody again. The transcriptomics said it would be more abundant. You can see here the images. This is the sweet localization in free-living dinoflagellate. It's the cell there, the chlorophyll in the nucleus. And you can see with the same sort of settings on the microscope, you can see vastly more immunofluorescent labelling in this cell, which is living inside the host. And then quantitated there. So these are 100 cells roughly counted, and the intensity of their staining. You can see in the free living situation, this is the fluorescence intensity, and the three separate experiments in the host at considerably more fluorescence intensity. So here, with the lack of the primary antibody, you can see that the controls are really quite low. And in fact, in the free living situation, there's not a lot more protein that we would have as the baseline of fluorescence in these. So vastly more fluorescence in these cells living in the host, which makes a lot of sense because that's when they need to export glucose. Whereas the free living situation, they'll be just pouring out valuable glucose into the ocean. To determine the substrate, we teamed up with researchers from just Gulf University in Malaria. They have a nice assay used complementation assay. They have a mutant EB15000 that is deficient for hexose transport, and it can still import disaccharides like maltose. So you put the genes that you want to explore the substrate for into an expression vector and then express those. So this base assay, the yeast will grow on maltose there, EB15000, and we've got several different variants. There's one transformer with a Arabidopsis thanauna sweet 1, which is a glucose transporter, and a Arabidopsis thanauna sweet 11, which is a sucrose transporter, and then dinoflagellate sweet 1, the Brugal sweet 1, and we also put in the empty expression vector. So these yeast are terrific for uracil. If you put uracil in the medium, they'll grow, but if you don't put uracil in the medium, then if they haven't got the expression vector, they won't grow. So obviously the untransformed EB15000 doesn't grow. So then you plate these into glucose and see what happens. And this is quite an exciting experiment because obviously the Arabidopsis one grows because it's got a glucose transporter and also a dinoflagellate one grew. So it tells us that this transporter is able to transport glucose. And there's a nice corollary experiment where you put in a toxic version of glucose, 2 grams of glucose, and if the cells are able to import that, they won't grow in this case. So the two lines able to import glucose are killed by the 2-deoxyglucose, whereas the other organisms are killed by that. So at this point, we had 3 nice bits of evidence. It's a bit like the game of Pluto, Professor mustard in it. Was a Colonel mustard in the library with a revolver or something like that. So the protein is in location, it's regulated at the right time in the symbiotic relationship. So it turns out that's the correct substrate. And then I sort of started scratching my head, what's the next experiment? And it's then you realise that you're working on a system that isn't as good a system as the plasmodium. We have no transfection system for these organisms, so we can't knock the gene down. We don't have any inhibitors for these particular transporters, so we can't sort of inhibit the transfer and see if that grows successfully. And we thought, okay, end of story, publish. And so we did. But this is how you generate a parasite. So dinoflagellates are sister group to apoptotic parasites, the phylum that includes Toxoplasma and Cryptosporidium and Plasmodium. And this organisms is sister group to ciliates, just in the broader evolutionary tree. And so we can assume here that photosynthesis was at this point here because the dinoflagellates are photosynthetic, and also because they're complex as well. Dinoflagellates make spectacular fossils. We know they're at least 500 million years old. We don't have no fossils for global complexes, but since they split from dinoflagellates, they use QED be 500 million years old as well. So I'll give a bit of history now. In 1996, Ian Wilson published a paper. This is a circular Wilson was sequencing through this and found that it had all of the same genes as a plant chloroplast. It was an algal chloroplast. The question is the DNA. And so it's kind of like fish, but instead of using fluorescence, you use gold particles. Okay? So what we have is and so So that's plastid, but it's not photosynthetic. It's a colourless plastid with no chlorophyll. And really, since then, the lab has been focused a lot on what does it do, and I'm not going to go into that today. It's quite a bit of it's so we see a little just full. So these organisms would have had symbionts. They would have had some sort of symbiont that was either mutualistic, parasitic, somewhere in between, because the symbionts pretty much everything has symbionts in them, etc. And so this is something that was. And so the symbionts was so how did they lose the photosynthesis but keep the plastic? Mutualists that cooperate with the host, the parasites then becomes so sweets are facilitators that transport the substrate but if the concentration gradient suddenly switches, low glucose situation here and a high glucose, the sweet simply reverses the flow of the glucose. Okay, and it occurred to me that this is what will happen if photosynthesis stops in these mutualistic situations. So now we know that the sweet from the dinoflagellate is bidirectional because we already set it up to be a glucose importer rather than a glucose exporter in the complementation assays. So in the flagellate, glucose goes outwards from the source by photosynthesis outwards. But in this, it has to actually come in from the from the agar plate to go into the yeast and then come and burn it and grow. So, so this sort of thing, well, when sweets and look at they're pretty much in all of these organisms except Cryptosporidium, curiously, which doesn't have an ADP. Maybe there's something going on there. But they're also in things called chromeras and cancers and that's significant as well. So I think sweets actually now are the first kind of molecular raising country for parasitism. And basically what would happen is that photosynthesis stopped in these mutualistic situations, and the symbiont then suddenly becomes a glucose thief instead of a glucose dominant. If that's true, then it created a whole flow of thousands of pieces of parasites. So it looks something like this. This is our symbiont, and it's photosynthetic, right? The plastic here, okay. So it has a sweet, which it only turns on when it's in the host, and then the glucose that's made by the plastic through photosynthesis is not exported when it's free living, but if that organism then moves to the host, then the glucose will be exported because the sweet will be upregulated, and this organism can proliferate in the host and export glucose and support its host in exchange for that precious nitrogen. So, membranes, sweet, as membranes we've shown, and that and so this is a win-win situation. Is now prospering from this mutualistic relationship. If the symbiont and somehow it loses its ability to photosynthesize, it's not first. Still obviously stealing nutrition from the animal host. Parasitism has evolved multiple times in this group. So before that, parasites here and there, but this group is passive, and it's actually more closely related to the parasites. Symbiotes you can reverse. So you can actually sort of put together and then they also have the ability to be living inside animal cells, intracellular cells. And photosynthesis in this image, the sweet just runs backwards. So in this sense, the sweet is actually complementing the mutation that breaks photosynthesis. So it's an instant rescue for that organism switching from a mutualist to parasite. And we get the same so this lineage is kind of the mother of parasites, where mutualists converted into parasitism. And all of that's very ancient, and I often tell my immunology friends that the reason you don't get immune to malaria is because they were there when the immune system was invented. They were dealing with the immune system when it was just innate, and the ancestors of malaria parasites are violent when the adaptive immunity was invented by animals. When this project back into the parasites and looking at the structure of the Plasmodium virus, this is the kind of flash, and this is the plant one, you can see that the parasite squid is also a very nice match to the plant one. A little bit of oxidation sentience in these helices, structurally looking very similar. Yet, the squid is dispensable in the blood stage, which I was a bit surprised about because we thought it's the reason for being a parasite in the first place. But this parasite also have a hexose, an active glucose uptake protein, so the capsules and it appears to be also dispensable in the mosquito stage, which is a bit of a conundrum because parasites don't have many genes that are totally redundant or dispensable throughout the entire lifespan. Yes. So I'm going to completely switch gears now. I'll get my drink of water and talk a bit more about a bit less basic science and a bit more sort of technological science. Who knows how the gene drive works? Don't be shy. Yeah, a couple. It's really interesting because they're really extraordinarily powerful things, and I think we all ought to be informed, and we all ought to be worried about them. They're remarkably powerful. And Matthew Cobb wrote this in his magnificent book that they are to worry about. But I think they were really useful. So gene drives give what's called super Mendelian inheritance. They're basically an unbeatable gene. So Mendelian inheritance is a 50-50 proposition. If these two genes, these two alleles reproduce together, heads and tails, the progeny will be 50-50. You'll get one head, one tail. And if those progeny mate with other progeny, doesn't get any more than the population, so the tails need to take over. But if it's a gene, say tails is a gene, the progeny of every class inherit the gene. Okay? So the progeny of this class will both be gene drives. Even if this mates with something else, they will come in our gene drive. Basically, every progeny is a gene drive every time. Okay? So that assures the powerful genetics equipment. The best thing is this idea of this prescribes that we make Chris using transposons and sensifiers use these to fix some of these things. It's fixing the population frequency. And so the idea of subsequent insecticide phenomena also enough of these mosquitoes. And so we can use it to eradicate the entire population. In this case, to eradicate this. When so and that's there's also 50-60 species. There's also a one built so yes, I said I guess it's a partnership. And it is, and we kind of complement it in lots of ways. And instead, we've got a plasmodium instead of mosquitoes. This is a bullet versus the bullet plasmodium. It has senses and for dendrites in that later. So reproductive cycle. This turns out to be a really, really big advantage for building and distributing a dendrite, which you don't have in mosquitoes. Predominantly, that makes the house just well, 50-60 species of these things. The downside so the ones that detect this was using single-sex parasites. So it's, as you know, plasmodium has an asexual cycle. It produces principally in the host and mouse around about 10 to 7 parasites. Some of those parasites sexual precursor forms, parasites, then to go into the mosquito. And asexual parasite can be into either a female parasite or a male parasite, so they're hermaphroditic. That are now known for those confusions. So, for instance, in this case, if the MD4 gene is expressed, that can either side that precursors sexual cell will become male gametes. And if you don't have the MD4 gene, then you won't get any male gametes produced in that particular asexual precursor population. There is female development, which is called the FD series, and if you knock out, for instance, FD1, then you don't get any females. And the line is then male. So what we did was just to test that this works. And so our system is plasmodium burgulae, infect a mouse with wild-type parasites. We then let a mosquito bite that mouse, and that mosquito becomes infected with the parasites. And then we look at the mosquito gut, and parasites and cysts mosquito. And we've got around 100 in our system typically. And then if those mosquitoes bite a naive mouse, the mouse gets malaria. Something wrong button. Okay. That's sick and could eventually die. If we looked at that native MD4 gene, then female development gene FD1, so basically, this is single sex parasite. So next what we can do is actually disrupt the MD4 gene instead of just a plain knockout if the gene drive cassette to the MD4 locus. So this is the MD4. Market DHFA in this case. So this is patients. So the experiment again is and this is the infection of the parasites. So so symbiosis. So let the gene drive go. So in this case, the gene drive parasite is an infection I showed you before. So that's FD knockouts. So the oocysts here. Now, all of the genotyping of these oocysts, they're all Cas9 positive and they're all MD4 minus. So the MD4 genes that came in from this line, even though they participated in the cross, the MD4 genes have gone. They've been destroyed by the gene drive. These mosquitoes will then bite a naive mouse and that mouse will get malaria. So the gene drive works and it's super efficient. In one generation, it was able to collapse the population. So then a nice control you can do is use a guide RNA that doesn't have a target. And so we switch out the MD4 guide RNA for a guide RNA that has a nonsense sequence, and then do the same experiment. And so in this case, this is the guide RNA that doesn't have a target in the Plasmodium berghei genome. And so we cross again the Cas9 gene-drive parasites, which is in place of the MD4 gene, with these guys which have the MD4 gene but no Cas9. Again, we get progeny, but you can see here that the MD4 gene has been transmitted into the progeny. It hasn't been eliminated by the gene drive. And these mosquitoes bite this mouse, it will get a malaria infection, and similarly, these mosquitoes bite this mouse, they'll become infected heavily with the Plasmodium. I got Drew to remake this video of the life cycle. So this is a mosquito after she's taken a blood meal. And we're going to see what happens with the gene drive. So in that blood meal, she takes up obviously lots of parasites, some of them asexuals and some of them gametocytes. And the asexuals get digested and go into being her nutrition. But the gametocytes are then activated, and there's a female on the left there and a male on the right. And the female, in our case, is the gene drive line with no MD4 gene. And so that female egg egresses, and then the male starts to exflagellate and produce the microgametes or the sperm, and it's an MD4 plus line. And this is one that Drew prepared earlier. And this little guy is coming in to fertilise this egg. He's about to get his nuts cut off. So he goes in, fertilises the egg, and that now becomes a diploid zygote. The male chromosomes come in, they merge with the female chromosomes as the two nuclei fuse during the zygote formation and syngamy. And what happens now is the magic for the gene drive, because these chromosomes are now preparing for meiosis to go in, because it immediately goes into the haploid phase again. And so the sister chromosomes line up, and these synaptonemal complexes here, which Drew's illustrated rather beautifully, bring the two sister chromosomes into register so that you can get meiotic crossover. And he's showing here now the DNA, and then we'll sort of pan out to show what's actually happening at the gene level. But this is great because it actually brings the two chromosomes lined up against each other right in register. And so all of the genes are all lined up together on this chromosome. So this is the chromosome of the target organism, which has an MD4 gene here, which is highlighted. And what happens now is the Cas9 has suddenly got a target. It didn't have a target in the gene drive organism because it's haploid and the gene didn't exist. And so the Cas9 will come along, identify the MD4 gene using the guide RNA and cut it. And these grey guys are actually the DNA break repair mechanism, the MDN complex. They're basically spooling up and down the DNA just in case there's a break. So Cas9 comes along, cuts at the MD4 site as guided. And these guys tether the two broken strands together. So the cell now has a crisis, broken DNA, and what happens is that corrections occur. So this will be a homology-directed repair. So this is chromosome 1, which has been cut by the Cas9. This is chromosome 1 from the gene drive organism. This is all homologous on this side and all homologous on this side. So it uses this segment to repair this double-stranded DNA break and copies the gene drive into where the MD4 gene was. So this zygote now is undergoing meiosis and turning into an ookinete, and that ookinete's got to get out of Dodge so it doesn't get digested, so it burrows its way through the blood and eventually gets to the gut wall, and then it punches a hole up through the gut wall where it can now relax and develop in the mosquito hemocoel and turn into sporozoites. So this is one that's going to come up through the gut wall, and it'll now gradually differentiate into an oocyst, and within that oocyst it'll develop thousands of sporozoites, all of which now carry the gene drive in the MD4 locus, and these sporozoites will now migrate somewhat mysteriously to the salivary glands and await for that mosquito to take another blood meal and deliver the gene drive organisms to the next vertebrate host. So there she is, loaded up with exclusively gene-derived sporozoites ready to infect the next host when she bites. The first time we told anybody about this, Hayley was one of the first people who said, but is it going to work in falciparum? You know, it's all very well in mice. I reckon it will. I'm 99.9% confident, but you never know. And we put our best postdoc onto this, who you'll recognise. It's kind of cool to have your academic grandchild come and work in your lab. So Mary Lou's building a falciparum gene drive, and with some help from Matt, who's worked up a bunch of single-sex lines, I think we're just about ready to do our first trial of the gene drive in falciparum. We did it with MD4 and FD1 because those were genes we knew and loved and had been working on for many years. This is an incomplete catalogue, Matt has now informed me, of all of the MD and FD gene series. There are a lot of them, and there's no reason that we couldn't target any of these genes, I don't think. And one of the things that Dean is building now is a gene drive in a male-only line. So we would deliver the gene drive to the eggs through the sperm rather than vice versa. It should work as long as the Cas9 and the guide RNA are expressed in those male gametes. So I think we can do this in lots of different organisms. You can do this, but should you?
Yeah, but your scientists were so
Oh, the sound didn't come out of my computer. I kind of like this, partly because Dr. Malcolm is not your usual scientist. We all know that it went really, really wrong in Jurassic Park. And so I've thought long and hard about all of this, often at 2 a.m. in the morning. There's a really nice model for how you would actually do this, because the mosquito gene drive people have also thought longer and harder about this for a lot longer time. And they've set up an organisation called Target Malaria. And when you look at what they're doing, a lot of the science seems to have been done. The building of the gene drive organisms is relatively complete. What they're now doing is addressing what do we do with these organisms. And this is a classic situation of sort of Western imperialism. We've got the solution for you, just stand aside. And they're not doing that. They're doing it quite differently. So this Target Malaria organisation have set up a whole network in Africa to build all of the social licence, the safety, the compliance situations to actually do a release based on the terms of the people who will be receiving this technology. And building that local compliance and getting the social licence and things like freely given informed consent with people who don't know what DNA is a really sort of, these are huge social challenges. And one of the things that Target Malaria do, which I particularly like, is they have these education campaigns. And they go out in the villages, and these two ladies here are facilitators, and their job is to go out and explain to the local people that this is what we're planning to do and this is how it will work. And I like this fellow here, he's put on his best hat for the day. And I like to think of this in two ways, this image, because I don't know what he's saying. He's either saying, take your bloody genetically modified organisms and go back to where you came from. Or he's saying, that's fantastic, my grandchildren are not going to get malaria because of the technology you're bringing us. And really, you know, both of those options are valid choices on his part, okay? So I'm now sort of trying to get together a team and some funding where we will look at getting this ethics approval, getting the safety coordinated, and James is on board to help out with some of those early trials in humans, and then figuring out with ethicists and people on the ground like Leanne Robinson, can we build freely given informed consent with populations and develop a social licence to use this? And then obviously there's huge local compliance issues. The consent bothers me most because a gene drive by nature is a drive, okay? You launch it here, it goes there inexorably. And so you get consent here where you release it, but what about in the village next door, or the country next door, or the continent next door? You can't build universal consent from everybody. So in some sense, there's a bit of a conundrum here of how to do that. We've done a bit of modelling. Ben Cam joined the lab about 6 months ago and has done some modelling. And we've looked at things like the efficiency. Is there a fitness cost to the gene drive? How many of a population would we need to inoculate to, say, drive the gene drive to saturation? How many bites are people getting? How many strains are people infected with? It's really quite complex. Ben was kind of overwhelmed initially, but he's built a number of models, and these are some populations of mosquitoes, I believe, 50,000 people and 30,000 mosquitoes. And then this is the time scale here, which is actually, he's put in egg laying cycles of the mosquitoes. So this is basically years to a decade here. And this is the gene drive allele frequency. So going from 0 before the gene drive is introduced to full saturation where all parasites have the gene drive. And this is a heat map where you start off with no gene drive in the humans and no gene drive in the mosquitoes, and then over this time cycle, and I'll play that now. So you can see that with an efficient gene drive and a low fitness cost, you get up to saturation or completion of the gene drive into the population within a relatively short period of time. And eventually all the parasites are gene drive organisms. And at this point, they would all be, for instance, female if we target the gene drive to MD4, and the population will then collapse. This is based on infecting 0.001 of the population. So not a huge number of people. It's still a lot of people, but you don't have to get to everybody and you don't have to get to everywhere. The natural system spreads it very efficiently. How would we actually deploy this? We thought a lot about this, and this is the best scenario we've come up with so far. I don't know if it's how it will actually work. So you would do it something like this. A cohort of patients presents at the local clinic where we plan to launch the gene drive with symptoms of Plasmodium, with symptoms of malaria. We treat them with a schizonticide, and their symptoms are resolved and they can go home. Now, at this point, they'd still be gametocytemic, which is sort of what happens most times anyway. A cohort of that population would volunteer to then receive the gene drive organism, okay? And so they would be deliberately infected with this GMO parasite. We would do that, we believe, using frozen stage 5 gametocytes. And gametocytes freeze pretty well. And so they would have to be developed in culture, and then they would be inoculated into these people. Need about 10 to the 5 gametocytes per person. So big lots of gametocytes, but it will be tractable. So these cohort would then also go home. They've already been treated with a schizonticide and they would have no symptoms, and they would be doubly gametocytemic. So our gene drive organisms would have a target line to mate with, because those people came in with Plasmodium already. A refinement of this strategy would be to give the people who didn't volunteer to be infected a gametocyticide as well, and they would then not be gametocytemic. We'd actually be reducing transmission. So the nice part about this strategy is that we're not creating more infections, and we're also not creating more transmission. Then these doubly gametocytemic people would be the target for mosquitoes, which would come along, bite them, take up the 2 organisms, they would mate, have sex, and the gene drive would then insert itself into the target population and spread further. We've come up with some other ideas for deployment, and I really like this one. So Plasmodium knowlesi is a zoonosis that spills over into people quite a lot in various parts of the Malay archipelago. Knowlesi uses Duffy binding protein alpha and the normocyte binding protein A to invade human erythrocytes. If you knock those 2 genes out, they can't invade human erythrocytes. So we could launch a gene drive that targeted those 2 genes using a multiplex guide RNA, 2 guides and 2 repair templates, to delete those genes, and then the parasite shouldn't be able to invade human erythrocytes, and we would eliminate spillover, whilst not changing the whole ecology of the Plasmodium knowlesi macaque infection, because there are some worries that if you take knowlesi away, the other infections like cynomolgi that they have will become quite severe. And this is an idea that's very much in incubation with Rob Moon and Yi-Ling Lau and Andy Waters to build. I will personally be supervising the deployment of this one along the Malay Archipelago, and I'm looking for crew. Keridan's already volunteered to be the bartender, but I do need a veterinarian probably to catch macaques and infect them. When you see these ideas, you'll think, what is he smoking? So one of the ideas that Dean came up with was to put a gene drive cassette with a cargo that would then express some kind of antigen that most everybody is immune to. So something like measles or polio or COVID, spike protein, for instance, so that you would immediately set the parasites up to be targeted by existing immunity. This has a problem that it will actually create a severe fitness deficit to the gene drive, and Ben tells us that this will slow down the drive. It will not go to saturation as quickly because everybody who's got the gene drive is getting hammered by people's immune system. But it's something we're talking to immunologists about at the moment. One I really, really like is to actually reverse drug resistance to drug sensitivity. Okay, so we could set up a gene drive, possibly with multiplex guide RNAs, to turn the chlorophyll resistance trait back to sensitive, to turn Kelch13 back to artemisinin sensitive, or to convert DHFRT, thymidylate synthase, back to sensitive. And this could be a multiplex situation where you could restore the efficacy, the sensitivity to multiple drugs with one gene drive. Again, if you did that, for instance, with the Kelch protein, you're going to have a fitness cost because if people are using artemisinin, the gene drive parasites that have been reverted to sensitive would actually be suppressed by artemisinin impression. So you might sort of split this up or something. We still need to look at does it work and is it viable? This, I think, is my favourite. So the idea would be to just knock out the VAR2CSA gene completely from the population. And this would, I think, in one swift stroke, prevent malaria in pregnancy, which is obviously a very serious problem, particularly because it impacts birth weight of the neonates, and that then puts them on the back foot for the rest of their life, basically, in places like Africa. So we could fairly easily build a gene drive just to take down that version of the PFEMP1, which allows them to colonise the placenta. With that, I'd like to thank Ashley, Karen, Giada and Madeline and Andreas for the work on the anemones and the dinoflagellates. And I've shown you this team here. Ton is our mosquito wrangler. Sophie developed the single-sex lines, the MD4 and FD1 knockouts, and Ben does our modelling. Thank you.
Thanks, Geoff. Take questions.
Thanks, Geoff. Very nice. I'm sure there's going to be lots of questions about the gene drive. I just want to ask quickly about the first half of your talk, the symbiosome. Is that a membrane?
Yeah, it's a membrane.
So how does glucose get across it?
Yeah, I don't know how that is dealt with. It's basically analogous to the Parasaurus vacuole. So it's created when the symbionts go in, but they're taken up by phagocytosis. So it's not, and the dinoflagellates don't seem to contribute any lipid to it. So I think it's all host membrane. Now the hosts then somehow decide, are they going to digest that food particle, or are they going to keep it as a friendly symbiont to manufacture glucose for them? And how they know the difference is not yet clear. But yes, it is another membrane. Glucose won't go across membranes easily.
Okay.
When I'm sort of, pipe dreaming, the hosts have a suite as well. And I really like the idea that they put their suite into the symbiosome to tap the glucose out from that vacuole space. But we've got to build an antibody and localise it, etc.
Geoff, that was incredible. I remember another quote from Jurassic Park, which was "Life finds a way." So with gene drives, is it I don't know, could you install a gene break as well in case of any ethics concerns where you can destroy your gene drive?
I like the idea of putting YCFU onto the Cas9, and we could then just simply give people, what is it called, 5-fluorocytosine, which is a licensed drug, and that would kill the gene drive in their parasite. So that's one strategy we have for a recall, and a fairly simple one. We don't have to, there's no technology to develop there. So that would be one thing. But yeah, life does find a way. And I mean, we've done a lot of work on drug resistance and selection, and you get resistance to gene drives. But it's more of a problem with things that have non-homologous end joining, because when they fix the break, they put sequence in there, and then the target sequence is destroyed for the guide RNA. So we got around that problem at least, but there will be, if the gene drive has any fitness cost, then obviously you start to induce selection against the gene drive.
I do remember there were papers about microhomology mediated end joining, MMEJ, in Plasmodium. Is that a concern at all?
Yeah. Yeah, it's possible. The beauty of the way the gene drive works is because the repair happens during meiosis, when the whole meiotic synaptonemal complex apparatus lines up your template in perfect register, and you've got the entire chromosome as homologous arms to affect the crossover. So it couldn't be any better as a mechanism of repair. Because it's happening in Plasmodium, it's great because they go into meiosis almost instantly after syngamy. So they exist as a diploid stage for maybe as little as a couple of hours. So it's the perfect organism to do this in, because there's very little time for repair to happen other than the crossover during meiosis.
Hi. Thanks. It was really a wonderful seminar. Lots to think about. And I think I also have to think about Jurassic Park, because they did something very similar, right? They created, I think, just one-sex dinosaurs, which is pretty much what you're trying to do. And I know it's really trying to predict maybe there might be genes that could create a resistance against, I guess, sexual stages. I mean, you can't predict that, but is there some kind of screen that you can do to see if there are particular genes that can be just slightly modified to get around the problem that there's just females? I don't know, CRISPR screen or something like that. Have you thought about? Because if there's just one gene that you can slightly modify to get around it, then it's not going to work, right? Because they might. Yeah,
you're right. No, we haven't thought about a screen. As I showed on that table from the Gockel paper, and as Matt Dixon has shown, there are a lot of these genes. There's a whole series of FD and MD. And I would imagine in the release or the deployed version, we would target more than one gene. So what we do there is you supply several guide RNAs, which you can drive multiplex. So you split the guide RNAs up either with a ribozyme between them or a tRNA sequence that's cleaved out after the polymerase generation of the RNA. And then you have to give multiple repair templates. So we might go for more than one FD or more than one MD, which makes it just more difficult for them to somehow get some sort of complementation factor to restore sex. The other thing I think could potentially happen is that the target population will start to self-fertilise more, that you would actually get selection for inbreeding in the target population. And I don't know what we can do about that. We're actually exploring now sex ratios. So Plasmodium generates typically more female gametes than males, gametocytes at least, because the male gametocyte generates 8 gametes and the female gametocyte generates 1. So if we could make a male vehicle gene drive and also change the sex ratio in the gene drive organism, we could actually kind of overwhelm the target population with lots of gene drive sperm. So that's on the drawing board. But whether that will work or not, can't say.
Last question.
So Geoff, just thinking about this, so the ethical conundrum here is that you need people to become infected with the gene drive so that it will then move through the population. And so the idea of a schizonticide is a good one because you can then suppress symptoms, but you don't want to give it too soon because you need people to make gametocytes.
Yeah. So
I'm wondering if there's, and you no doubt will have thought about this, with some of the emerging biology around gametocytogenesis, if there are, with talking to Matt or to Till or to others, genes that are known to kind of regulate when gametocytogenesis turns on. As you know, Vivax comes out of the liver with PFS16 on and becomes gametocytes almost instantaneously. So that would be great for a gene drive because you could not have the symptoms from the, you could almost gene drive away asexuals and just allow the gametocytes to go. For falciparum, you have to think about what could you do genetically to basically suppress asexual parasites and allow gametocytogenesis to happen quickly.
Yeah. So thinking about Till's line with the inducible gametocytemia, that was something we thought about building in, and the gene drive guys would be sexy early, and so they'd have the drop on the target line. The more of these things we sort of think we'll build in, it becomes more and more complicated and complex, and so there's a tendency to want to keep it simple and lean versus add bells and whistles. And so I think, we'll have to find the balance for that. But you're right, that would give the gene drive an advantage.
It might still be
You've got to all link it all up to cargo in the gene drive though, because we thought of also making the initial infection organism, making it less virulent. We take away a whole bunch of things to make it less virulent. But everything's getting crossed over anyway during that meiosis. So the progeny will be a mashup of the two parentals. So we'd get away with it for a generation or two, but eventually it'll be, whatever the progeny is selected for in people. And probably the parasite needs to be reasonably virulent to be competitive. It's got to survive in people, reach parasitemia that's high enough to generate gametes, etc. So, we can, and all of it's got to go into the gene drive cargo locus, and with multiplex guide RNA. So more parts, more complexity, more opportunity for fail or dysfunction.
All right, we're after 2 o'clock now, so that just remains to thank Geoff for a great seminar.